everyone. Today my group and I will be introducing the tardigrade and how it has been used as a non-traditional model organism. The tardigrade is a segmented, extremotolerant microorganism that makes up the Tardigrade phylum and is related to both nematodes and arthropods. The Tardigrade phylum consists of over 1,300 distinct species that have currently been identified, although researchers do speculate that there's plenty more that we just haven't found yet. Additionally, tardigrades are over 500 million years old, and they have survived all 5 mass extinction events on Earth, and this makes sense because tardigrades are most well known for their cryptobiotic abilities, where they're capable of surviving extreme temperatures, extreme pressures, a complete lack of oxygen and or water, high doses of radiation, high concentrations of toxins, starvation, cosmic vacuums, and extreme speeds and impacts, and they do all of this by entering a mostly metabolically inactive state called a tun state. And because of this, tardigrades are super adaptable and are found on all continents in both aquatic and terrestrial conditions. And they're often seen to be living amongst moss, hence the moss piglet nickname that they've been given. The tardigrade is a very unique model organism, mainly because of their cryptobiotic abilities, and there's been a lot of research into the exact mechanisms of how their cryptobiosis works, and there have been some significant findings, but much of the mechanisms are still unknown. Some of the suggested mechanisms pertain to how the tardigrade is capable of repairing DNA damage from radiation exposure and their use of tardigrade-specific intrinsically disordered proteins, or TDPs for short, and my group mates will go over that a little bit later. The tardigrade is a useful model organism because they're relatively adaptable to the lab setting, so they share similarities to C. elegans and Drosophila, and they have a short generation time, a compact genome, highly reproducible developmental patterns, and they can be cryopreserved for long periods of time. So this graphic here depicts cryptobiosis and the extent of the extremes that tardigrades are capable of enduring. So on the top you see tardigrades' active state, where they're able to grow, eat, and replicate like normal. And on the bottom, you see the tardigrades' tun state after they've been exposed to some extent of environmental stressors, where their metabolic rates are now 0.01% of their normal metabolic activity, and their body shrivels up to a third of its typical size in response to either dehydration or freezing. The tardigrade has been used as an animal model in the fields of astrobiology, genetics, adaptive evolutionary biology, developmental biology, and biochemistry, and my group mates will explain some of the examples of how tardigrades have proved to be useful model organism in some of these fields.
Tardigrades have been the subject in body-plan evolution studies. The patterns of their expression of Hox genes show similarity to the patterns of arthropods and other animals. The study of the development of the tardigrade body plan has also given different insights as to how common ancestors divide and diverge into new, unique organisms. In different astrobiology studies, extremophiles were used to study DNA damage and repair mechanisms. Tardigrades were perfect for these studies due to their relatively complex structure, resiliency to extreme environmental stressors, and their ease of observation. During the Photon M3 mission studies of 2007, 3 projects were conducted on tardigrades. These studies exposed both active and inactive tardigrades to radiation in microgravity conditions, the vacuum of space, and other extreme stressors. The TardiKiss project had similar goals as well. Some of the results from these studies revealed that the survival rate of tardigrades was not greatly affected by microgravity and cosmic radiation, and that tardigrades can survive exposure to the vacuum of space. The exact mechanisms that give tardigrades this great protection from DNA damage are not fully known, but give much to be explored. One thing that scientists believe could contribute to the resiliency of tardigrades are intrinsically disordered proteins, or IDPs. In tardigrades, these IDPs are called tardigrade disordered proteins, or TDPs. Unlike regular proteins, the function of these TDPs are not dictated by their 3D structure. Rather, their structures are not static and have dynamic and changing properties. These TDPs have shown to be crucial in the protection from desiccation of tardigrades, and the genes of these TDPs may provide more knowledge on disordered proteins and their involvement with stress tolerance.
Certain tardigrade species express proteins that are capable of protecting a specific enzyme in drugs such as vaccines. These specific enzymes are called biologics, and the tardigrade proteins protect them from freezing or desiccation. Many tardigrade species are thought to express these proteins, which are called cytosolic abundant heat-soluble proteins, or CAHS proteins, except for the marine species of tardigrades. CAHS proteins are a type of intrinsically disordered proteins, which are also called IDPs for short. And, as mentioned earlier, the IDPs are referred to TDPs in tardigrades. Since TDPs lack a well-defined three-dimensional structure, unlike the more well-known rigid proteins that most of us are used to talking and learning about, they offer some advantages. Upon exposure to dry environmental conditions, these specific CAHS proteins undergo transformation. Beginning as a shapeless and flexible type of protein, they transition into a type of bioglass, and this is capable of holding specific molecules and proteins together until the tardigrade is in more suitable environmental conditions. In addition to their potential use for preserving molecules used in vaccines without refrigeration, as well as their potential use in improving crops' resistance to drought, IDPs are also associated with a number of diseases. Some of these diseases include Alzheimer's disease, cardiovascular disease, cancer, and diabetes. Understanding how these proteins function can further improve general scientific knowledge of TDPs and their roles in these diseases. Tardigrades express a protein called DSUP, which stands for damage suppressor. This protein is a type of TDP, and it is discovered to prevent tardigrade DNA from radiation and desiccation-induced deterioration. Researchers discovered that altering cultures of human cells to make them express this protein increased resistance to damage induced by X-rays by 40%. This has major implications, such as protecting patients going through chemotherapy. Since DSUP also protects DNA from damage by hydroxyl radicals, this has implications with the genomic instability problems associated with both cancer and aging.